Hello, everyone. Welcome to today's live broadcast, Structural Characterization of Biopolymers by Analytical Separation Techniques with Advanced Detectors. I'm Laura Bush, the Editorial Director of LCGC, and I'll be your moderator for today's event. We're pleased to bring you this web seminar presented by LCGC and sponsored by Wyatt Technology Corporation. I would now like to introduce today's speaker. We are very pleased to be joined today by Professor Stepan Podzimek. Professor Podzimek, thank you for joining us today. Please get us started.
Hello, everyone. It is my pleasure to present this webinar that is devoted to the characterization of biopolymers by size exclusion chromatography or asymmetric flow field flow fractionation coupled to multi-angle light scattering detector and possibly also to online viscometer. Here are the topics to cover. I will briefly remind the basic principles of the techniques that can be used to characterize biopolymers, and then I will focus on the examples of characterization of 4 different biopolymers materials. Concerning analytical separation techniques, we have 2 possibilities: size exclusion chromatography and asymmetric flow field flow fractionation. Size exclusion chromatography is also known under its original name, gel permeation chromatography. It is well established and widely used analytical separation technique that is used in all fields of polymer science to determine molar mass distribution. The main separation mechanism is called steric exclusion. The molecules of different size permeate into the pores of porous column packing. The smaller molecules can permeate deeper into the pores, and larger molecules are excluded from certain volume. In the ultimate case, the biggest molecules may not permeate into the pores at all. That means that the separation is based on the hydrodynamic volume. The molecules with the highest hydrodynamic volume elute from the columns first. They are followed smaller and smaller molecules. The picture shows example of separation of 4 narrow polystyrene standards. This slide shows a typical SEC MALS system. On the left-hand side, there is HPLC system. It can be from any manufacturer. Typical parts are degasser, pump, autosampler, optionally UV detector, the applicability of which are limited in the case of biopolymers. Then we have columns, usually 2 or 3 columns are used in series, and then the system of detectors. The most important detector is multi-angle light scattering detector DAWN. Optionally, we can use online viscometer Viscostar, and the last detector in the series is refractive index detector Optilab. The signals from detectors go to PC with the software Astra that acquires the data, and then they are later on processed. Asymmetric flow field flow fractionation is less known and less established than traditionally used SEC. Anyway, in the past years, it is becoming more and more used as it offers several advantages compared to size exclusion chromatography. Separation is achieved by flow of sample molecules through thin ribbon-like channel that is created by sandwiching a thin spacer between the two blocks. One block is solid, the other block is semi-permeable, created by a frit that is covered by semi-permeable membrane. A typical material for the membrane is regenerated cellulose. The solvent, in the terminology of AF4 called carrier, comes from HPLC pump and flows through the channel in two directions. The first direction is from channel inlet in longitudinal direction to channel outlet towards detectors. The second flow is in cross direction as the wall is permeable for the solvent. The separation principle is explained on this scheme. This is solid wall, semi-permeable wall. The distance between the two walls is given by the thickness of the spacer. Usually it is 350 micrometers. The sample molecules are pushed by the cross-flow field towards the semi-permeable wall. They cannot escape as the wall is permeable only for the solvent. The counteracting force is diffusion. The smaller molecules have higher diffusion coefficients, and thus they can counteract the cross-flow field more, and they create a cloud which protrudes more from the wall compared to the cloud of larger molecules. It's lower diffusion coefficients, and as they cannot counteract cross-flow field, they are more compressed to the membrane. The flow profile in the channel is parabolic. The highest velocity is in the channel center, and the velocity decreases towards the wall. The thicker clouds of smaller molecules protrude more to faster moving streamlines, and that is why they elute from the channel before the larger and larger molecules. So the separation is in opposite order compared to size exclusion chromatography, that is from small molecules towards larger molecules. The example shows separation of 6 narrow polystyrene standards. The difference between the two separation techniques, so they are very similar. They use the identical HPLC instrumentation, identical detectors. SEC is actually the true chromatography because the separation is achieved by creating equilibrium between two phases, between stationary phase and mobile phase. AF4, strictly spoken, is not a chromatography because the separation is achieved only in one phase, that is mobile phase. The picture shows a typical AF4 channel compared with SEC column of regular dimensions, 300 millimeters in the length, 8 millimeters in inner diameter. Particle size is usually 5 micrometers. Next slide shows a scheme of AF4 system. Again, it consists of HPLC part, degasser, pump, autosampler. The column is replaced with the channel that is connected to the controlling and measuring unit Eclipse. And in this example, we have only two detectors, multi-angle light scattering detector Dawn and RI detector Optilab. So we have separation techniques and we need detectors, and we are talking about advanced detectors. So advanced detectors, it means that these detectors are able to directly measure important characteristics of polymer materials. So these characteristics are most of all molar mass, and in addition root mean square radius, hydrodynamic radius, intrinsic viscosity. To get these quantities we need detectors. They are multi-angle light scattering, abbreviation MALS. It can be embedded with dynamic light scattering. And the third detector is online viscosity detector. Let me start with multi-angle light scattering. It is the technique that measures the intensity of scattered light simultaneously at multiple angles. So the scheme of the instrument is shown here. Incident laser light, and the flow cell is surrounded by the array of photodiodes that measures the intensity at different angles. This slide shows basic light scattering equation that relates the quantity that we can measure, that is Rayleigh ratio, with the quantity that we want to measure, which is molar mass. P theta is so-called particle scattering function. At zero angle it becomes unity, and it also allows to determine root mean square radius. C is the concentration, K star is optical constant that includes optical constants like refractive index of the solvent, wavelengths of the light, DNDC, specific refractive index increment of the polymer that we measure in given solvent. A2 is the second virial coefficient, and at low concentrations typical for SEC or AF4 separation, this term can be neglected. This slide explains how the molar mass is measured by multi-angle light scattering. So the intensity is recorded at different angles, extrapolated to zero angle, where the particle scattering function becomes unity. I already mentioned that the term with the second virial coefficient can be neglected. That means that after simplification, the equation can be used to get molar mass from the Rayleigh ratio at zero angle and the concentration that is measured by concentration-sensitive detector, mostly refractive index detector. The slope of this plot allows calculation of root mean square radius, also called radius of gyration, as important additional piece of information. Intrinsic viscosity, it is also called limiting viscosity number. It is specific viscosity divided by concentration at concentration approaching zero. Classical determination of intrinsic viscosity is using capillary viscometers when the sample is prepared at several concentrations, and each solution is measured with the capillary viscometer. The measured quantity is time that is needed for polymer solution to flow between the two marks of the viscometer. Specific viscosity is calculated from the time, and then the plot is specific viscosity divided by concentration against concentration, and by extrapolating to zero concentration one gets intrinsic viscosity. online determination of intrinsic viscosity is its online viscometer. The main part of the viscometer is capillary bridge that consists of 4 capillaries, delay volume, and 2 pressure transducers. One pressure transducer measures the pressure difference between the 2 lines of the bridge. The second one measures the pressure difference between the inlet and outlet of the bridge. The specific viscosity is then determined from the pressure, and as in SEC, the concentrations are very low. We can neglect the concentration dependence, and the intrinsic viscosity is calculated from the specific viscosity divided by the concentration, and this is done at regular time intervals and as in the case of molar mass. Bio-based polymers, they can be separated into several groups. The first group are polymers that can be found directly in the nature. Typical example is cellulose or starch or lignin, also natural rubber that can be also made synthetically, as natural rubber is nothing else than polyisoprene. The second group are polymers that are made by synthetic procedures, but at least partly from bio-based materials. These polymers are various derivatives of cellulose, like nitrocellulose or cellulose acetate, and then other polymers that often include vegetable oil. Then we have the third group of polymers that are made by microorganisms. Example in this case would be hyaluronic acid, well-known material from its application in pharma and cosmetic products. And finally, we have polymers based on biomonomers. That means they are polymerized by synthetic procedures, but from the monomers that can be made by bio processes. Example in this case is polylactic acid, as lactic acid can be produced not only by classical way from crude oil, but it can be also made by bacterial fermentation of sugar. Concerning the characterization of biopolymers, so I'd like to emphasize that generally there is no principal difference between the analysis of synthetic polymers and natural polymers. We use the same instrumentation and we want to get the same kind of information. However, at the area of biopolymers, we can encounter several difficulties. First of all, it is limited solubility. Cellulose is a nice example, or starch. To overcome the solubility issue, often we have to use highly polar solvents like DMSO, DMSC, DMF, often with the addition of salts like lithium bromide or lithium chloride. Sometimes we have to use several steps of pretreatment, like soaking the sample in various solvents before dissolution. Or we can also derivatize the samples to make them soluble in THF, because THF is easy solvent for SEC or AF4. That means example can be cellulose tricarbonylate or acetylated lignin. Another possible issue is fluorescence of some natural polymers that concerns lignin and humic acids, and when fluorescence is not compensated for, it results in strongly overestimated molar masses. Finally, There are no standards well described in concerning molar mass and molecular topology. So molar mass is not issue because we measure molar mass in absolute way by multi-angle light scattering detector. But when it comes to branching, the branching studies are based on comparison with linear counterpart, and typically we don't have any linear model for biopolymers. So application examples. So I will start with polysaccharides. This slide shows Markov plots, that is relations between intrinsic viscosity and molar mass for 5 different polysaccharides measured in aqueous buffers. And the slopes and the patterns of Markov plots can reveal information about the structure of the polymers. The green plot is for pullulan, so it's perfectly linear with the slope 0.62. So 0.6 to 0.8, these slopes are typical for linear polymers. So the slope and the linear pattern of this plot corresponds to linear macromolecular chains, and really pullulan is linear polysaccharide. Then we have another plot for dextrane in red color. So dextrane is known as branch polymer, and branching here is indicated by curvature of the plot, which decrease in slope towards higher molar masses. So this is typical shape for branch polymer. And then we have 3 other polysaccharides with the slopes, at least a part of the plot has the slope that is close to 1. And the slopes 1 and more are typical for extended rod-like structures. And indeed, these 3 polysaccharides are polyelectrolytes, and we can expect that the chains are expanded by electrostatic forces. So, and then all 3 plots show at certain molar mass deviation from linearity. So the slope decreases. In this case of blue polysaccharide, it even comes to 0. And this decrease in slope indicates more compact structure that can be explained by presence of branch macromolecules or by coevolution of aggregates. Next slide shows example of the analysis of cellulose. In this example, the cellulose was dissolved in DMAC after pre-treatment in various solvents. The molar mass across the peak decreases evenly, which gives evidence about efficient SEC separation. And at this point, I can say that we measured 2 similar samples. And when we compared their distribution curves, they almost overlapped. That means that the first conclusion was that the samples are identical. But with multi-angle light scattering detection, we get additional information, which is root mean square radius, and then we can plot root mean square radius against molar mass in double logarithmic scale. The plot is called conformation plot. And if you look at the conformation plots of the 2 cellulose samples, we can see the difference. The blue sample has the slope of 0.57, that is typical value for linear macromolecules. The red sample has markedly lower slope that indicates branching of the polymer chains. This slide shows one more example of the analysis of cellulose. So in this case, the sample was derivatized to tricarbonylate so that we could measure it in THF. And the separation in this case was achieved by AF4. The refractive index signal shows a strong peak at the region of lower molar masses, so this peak can be assigned to hemicellulose, and then we have another peak at the region of higher molar masses that corresponds to cellulose. The question is, why to use AF4 instead of SEC? So the question is answered in the next slide. That compares distribution curves of the same sample obtained by SEC moles in red and AF4 moles in blue. So distribution obtained by SEC is markedly narrower compared to AF4. The curve is shifted to lower molar masses at the high molar mass part of the distribution, which is also evident from the weight average molar mass that is lower when measured by SEC. On the other hand, in the lower molar mass part of the distribution from SEC is shifted to higher molar masses, and again it is evident from the comparison of number average molar mass, which is markedly higher in the case of SEC. The explanation here is that the sample contains branched macromolecules that can be anchored in the pores of column packing, and due to the anchoring they elute later at the end of chromatogram together with smaller molecules. And as the light scattering detector is sensitive mainly to the molecules with high molar mass, so these high molar mass delayed fractions actually overshadow the smaller molecules. And for polydisperse elution volume slices in SEC, the mass detector measures weight average molar masses, and actually the distribution is falsely shifted to higher values. In the region of high molar masses, the shift of distribution obtained by SEC can be explained by shearing degradation in SEC columns. And this shearing degradation is eliminated at least partly in AF4 because typical pressure in SEC columns is several times higher compared to the pressure in AF4 channel. That means that SEC actually fails to separate such sample properly, and AF4 is evidently more appropriate and more efficient separation technique for such samples that contain fractions with very high molar mass and also branched molecules. Polylactic acid and its copolymer with glycolic acid is material with various applications. One of them is using as drug delivery system, and PLGA can be synthesized as linear or branched polymer. And branching is one of the parameters that can be used to control the drug release rate. And then it is important to have analytical tool that is capable of characterizing branching. Such analytical tool that can characterize branching is a combination of SEC with MALS and online viscosity detectors. They provide data for Markov plots, as already shown for various polysaccharides. So here we can see the Markov plots of linear PLGA in blue. The plot is linear, the slope 0.55. It corresponds to oligomers because linear oligomers have the slope of 0.5, and we are in the range of oligomers. Then we have 3 PLGA samples containing different amount and 2 different types of branching agents. And the technique proves the presence of branch molecules, and it can also differentiate different degree of branching. The sample in green is, in this case, less branched. On the other hand, the sample in magenta is the most branched polymer in the investigated series. Lignin and similar materials, humic acids. Lignin and humic acids are the materials that require highly polar organic solvents to be dissolved. Alternatively, they can be transferred to derivatives soluble in THF by acetylation. The issue in their characterization is fluorescence that results in significant overestimation of molar mass. And later on I will explain how to reduce the effect of the fluorescence. What are the symptoms of the fluorescence? The first symptom of the fluorescence is decrease of the signal of forward laser monitor. Forward laser monitor is one of the unique features of Wyatt technology MALS detectors. It is the detector that is placed after the flow cell, and it monitors the intensity of light going through the cell. In the case of samples that absorb incident light, one can see the decrease of the forward monitor signal, which is associated with the signal of RI detector that corresponds to the concentration. Near the apex of RI signal, we have the minimum signal of forward laser monitor. Anyway, the decrease of the intensity recorded by forward laser monitor does not necessarily mean that the sample is fluorescent. We can have also samples that absorb incident light and they are not fluorescent. Anyway, without correction for the incident light absorption, we would get incorrect molar masses, and the absorption of the incident light by sample is corrected by Astra software, and the correction allows the determination of the correct molar mass. Next slide shows that different samples can absorb light with different intensities. So this is example of forward laser monitor recorded for 4 different lignin samples prepared using different procedure. And the sample in magenta absorbs just slightly. On the other hand, the sample drawn in blue color absorbed with very high intensity. That means that in this case, at this concentration, the intensity dropped almost to 0. Another indication of fluorescence is suspiciously high molar mass and unusual molar mass versus elution volume plot. Typically, the molar mass decreases at the very beginning of chromatogram, but then it increases again and reaches the maximum close to the maximum of RI signal. That means close to the maximum of concentration of eluting molecules. Then it goes down again, but even at the end of chromatogram, it stays at very high values. So this is another evidence of possible fluorescence. And final evidence can be obtained by comparing the signals recorded by photodiodes that are covered with interference filters with the signals from photodiodes that are not covered by fluorescence filters. So if we can see such strong difference between photodiodes covered with filters and photodiodes without filters, this is clear evidence of fluorescence. This slide summarizes how to deal with the fluorescence issue. So the users that already have MALS detector with regular laser wavelengths can only use fluorescence blocking filters. And these filters are mostly not enough. That means that researchers that are more involved into the research of lignin and similar fluorescent materials need MALS detector with infrared laser, and this detector is optionally available from Wyatt technology. And even for infrared laser, I would highly recommend to buy also several fluorescence blocking filters. Not necessarily on all photodiodes, quite the contrary. If we have photodiodes that are covered with the fluorescence blocking filters and several photodiodes that are not with these filters, we can investigate the intensity of fluorescence. This picture shows analysis of highly fluorescent lignin. The molar mass versus elution volume plots recorded with different instrumentation. First of all, the results obtained with regular laser with no filters. So the molar mass after decrease goes up again to very high value, around 1 million, and then decreases again, but even at the end of chromatogram, it stays at suspiciously high value. Then we have in blue regular laser with filters. They help, but still the effect of fluorescence is very strong. Using infrared laser without filters, the results are shown in green. So we can see strong reduction of fluorescence by going to infrared laser, and we get even more reduction of fluorescence when infrared laser is combined with fluorescence blocking filters. The previous example showed that for highly fluorescent samples, even using infrared laser and filters, may not reduce fluorescence completely. For such samples, we can use the procedure that is shown on this slide. And the idea of this procedure is given by the fact that at the beginning of chromatogram, the concentration of elutin molecules is low, and such is the fluorescence, as the fluorescence intensity is directly proportional to the concentration of molecules. On the other hand, the intensity of scattered light is directly proportional to the concentration and molar mass. That means that the intensity of scattered light at the beginning of chromatogram is high, and the scattering intensity overwhelms the fluorescence. Due to this fact, the molar mass data at the beginning of chromatogram are correct. They are not affected by fluorescence, and it allows extrapolation of the molar mass versus elution volume plot forward to the end of chromatogram, where the effect of fluorescence is not negligible. This way, we can get true molar mass distribution and molar mass averages that are not affected by fluorescence. The effect is mainly on the number average molar mass, as shown on this table. So we can see that using extrapolation results in significantly lower number average molar mass. On the other hand, the effect on weight average molar mass is not that evident, as the weight average molar mass counts mainly the fractions with higher molar mass that are not affected by the fluorescence. The last example is for polymerized vegetable oils. They can be used as environmental friendly lubricants. Example of the analysis of polymerized vegetable oil is shown in this slide. The signal from RI detector shows intensive peak. Its corresponding molar mass below 1000. So this is the peak of starting oil. And then we have the signal from polymerized molecules and their molar mass. The cumulative distribution plot that we can get from the acquired data describes the fraction of unreacted oil, so the fractions below about 1000. So in this particular case, about 30% of sample has molar mass below 1000. So these are the fractions that are not polymerized, and then we have the products of polymerization reaction and distribution of mass in these products. In addition, for this sample, we also used online viscometer, so we could get Markov plot nicely linear, but the slope was 0.3. That is much below what we would expect for linear molecules, and this gives evidence about presence of branch macromolecules, which makes sense because each oil molecule contains several double bonds, and that means that when these double bonds undergo polymerization, the resulting polymer molecules are branched. Based on the presented results for various biopolymers, we can conclude that online mass and viscosity detectors can provide not only absolute molar mass distribution, but it can also provide interesting information about the molecular structure of various biopolymers. Fluorescence of lignin and humic acids can be addressed by using Wyatt technology, Dawn Mal's detector equipped with infrared laser and fluorescence blocking filters. Asymmetric field flow fractionation, which is also available from Wyatt technology, was shown as an alternative separation technique that finds utilization mainly for biopolymers containing fractions with very high molar mass and especially branch macromolecules. Thank you for your attention, and I will be happy to answer your questions.
Thank you, Professor Podzemek, for that informative presentation. It is now time for the question and answer period. Here's a first question. What is the difference of conformation estimation by using MH plot versus using molar mass and RMS plot?
Okay, so this is a very interesting question. So generally, both plots should provide similar results. They should identify branching, and they should allow distinguishing between different samples. The advantage of Markov plot is that is more sensitive to branching, as the slope of the plot is in the range of 0 for highly compact molecules, up to more than 1 for extended chains, as we could see on one of the examples. And also another advantage of Markov plot is that it can be also used for smaller polymer molecules, whereas root mean square radius can be measured down to about 10 nanometers, which is roughly 100,000 gram per mole molar mass, and below that cannot be measured.
Thank you. Here's another question. What is the molar mass range for AF4?
So I will start from at the region of high molar masses. It's generally unlimited. The highest molar masses I measured were up to 100 million gram per mole. In the region of lower molar masses, the limit is given by cutoff of the membrane in the channel, which is usually in the range of 5 to 10,000 gram per mole.
Thank you. This next question is a little bit long, so I'll read it to you, and you can ask me to repeat any of it if needed. I'm familiar with a recent publication in which you are a co-author about absolute molecular weight characterization of lignin using SEC-mols. One question I have about it is why Ri is used for lignin as the concentration detector, given that UV absorbance at 280 nanometers generally gives a much stronger signal. Is there a benefit to using Ri over UV in this case?
Yeah, so of course you can use UV detector as well, but the UV signal can be even too strong and it can get saturated. So I would always prefer RI signal, RI detector, which is more general, and yeah, that would be, yeah, I would always use RI signal, but UV can be used as well, of course, as a concentration detector.
Okay. What is the advantage of using a viscometer for the characterization of branching?
Yeah, this is a similar question to the first one. As I already said, it is more sensitive because we get Markov plot, the relation between intrinsic viscosity and molar mass. We learn from the slope of that plot for highly compact, highly branched macromolecules, it can drop down to 0, and it goes more over 1 for extended structures. And the second advantage is that it also can be used in the region of oligomers. The applicability starts at just several hundred grams per mole.
Thank you. How does the LSD give the molar mass of molecules, and how do you compare that to mass spectrometry?
So, comparison to mass spectrometry, it can be only in the region of lower molar masses. I think that if we are in the range of hundreds, thousands grams per mole, mass spectrometry cannot be used. So that is probably the main advantage of light scattering, if this answers to your question.
Thank you. For polymers that are not soluble and highly cross-linked, would the FF4 technique be suitable for the determination of molecular weight?
So if the polymers are highly cross-linked, and that means that they create some supermolecular structures where we have more macromolecular chains than just single chain, then SEC cannot be used at all. And if these species are still dispersible, if they create, let's say, some type of nanogels, then they can be separated by AF4, and the response in light scattering is very strong, so they can be nicely measured. And of course, when it comes to highly cross-linked structures, we still can measure molar mass, but actually it is not molar mass because we are not working with molecules anymore. We are just dealing with some supermolecular structures that some people call microgels. I would call it rather nanogels because they are in the size of several hundred nanometers. But AF4 would work for sure if you have such materials. If we have macroscopic gels, then they cannot be measured by any separation.
What are possible solvents for AF4?
Once again, what are possible solvents?
What are possible solvents for AF4?
Okay, so for AF4, we can use aqueous buffers. That is many times case of polysaccharides. We can also use organic solvents. DHF works very well, and it also dissolves many polymers. Other solvents that can be used include dichloromethane, chloroform, toluene. I also tested. So generally, except for DMSO that would dissolve semi-permeable membrane of the channel, you can use most of the organic solvents. As I already said, all aqueous buffers.
Would you recommend analyzing cellulose in its native form or after derivatization?
Yeah, this is a very nice question. So, of course, that the tendency is not to treat that much the polymer before analysis, because any derivatization brings certain possibility of changing that polymer because we change the structure. On the other hand, cellulose can be pretty difficult to dissolve, even if we use strong organic solvents like DMSO or DMAC. And sometimes we may not be able to dissolve it properly. Then derivatization helps a lot, and especially if we make cellulose tricarbonylate, then it is well soluble in THF, and then it can be nicely measured. So I don't have 100% recommendation. So if you find the way how to dissolve, then I would try to avoid derivatization. But many times it is impossible, and then derivatization helps.
How can dN over dC be measured?
Oh, dNdC can be measured either so-called online. That means we need to know the concentration of injected sample. We also know the injected volume, which means we know the injected mass. I mean the mass injected into SEC columns. And then Astra software allows to determine dNdC from the injected mass and the peak of RI detector. This works very nicely. The only assumption is that we work with pure polymer and that we can assume 100% mass recovery of the injected sample from the columns. Alternative way, which is more laborious but also well feasible, is that we prepare the sample at several concentrations covering about 1 order of magnitude. The solutions are injected by hand directly into RI detector. And again, Astra software has a method to measure dNdC by this way. So, and these two procedures are very well comparable.
Can you utilize a viscometer to characterize the difference in density of similar biopolymers?
Yes, it can, because viscometer measures intrinsic viscosity. Intrinsic viscosity, the unit is milliliter per gram. That means that it is the volume of the polymer coil related to the mass. That means that actually it is reverse density, but we are talking about density of polymer coil, not about the bulk density of material as it is. Yes, it can be used for this.
Great. One last quick question. How would you recommend monitoring reactions such as polymerization or depolymerization?
Yeah, so I would recommend SEC in combination with multi-angle light scattering. It can be done very quickly. You can also use now APC columns that allow separation of polymer sample just in a few minutes. And also Wyatt technology offers detectors for APC. And then you can get information about the molar mass distribution just in a few minutes, and it is well suitable to monitor the polymerization reaction. Concerning depolymerization, that's probably not, you don't need the information that rapidly, but again, it would be SEC mouse. Probably you don't need APC and micro detectors, so you can use regular SEC columns and regular line of detectors. So mass detector would be the most important. And if you want also to follow the branch in development, then of course viscometer would be also recommended.
Fantastic. Well, we're out of time. Professor Pozemek, thank you very much for your talk today and for answering all those questions.
Yeah, thank you also for your attention.
Yes, and I want to thank everyone in the audience for attending and participating in today's event. And I would like to thank our sponsor, Wyatt Technology, for making today's webcast possible. Everyone in the audience will receive an email from LCGC alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.